Hi, this is Kavita Kundra and welcome to Mscdfsm Biochemistry class, chapter name Enzyme and Coenzymes. Enzymes. They are the protein that catalyze biochemical reactions. Louis Pasteur demonstrated the fermentation of glucose by yeast and catalytic agent was called as ferment, and then Buchner and other found that cell-free extract of yeast can ferment glucose. 1926, James Summer revealed the true nature of enzyme as he extracted the urease from jack beans. At present, 150 different enzymes are known. Chemical nature of enzyme. Enzymes are high molecular weight compounds made up of amino acid linked by peptide bonds. They have molecular weight from 10,000 to 2 lakhs. All proteins are enzymes and RNA can act as enzymes. Such RNA are called ribozyme. So the ribozymes are RNA molecule with catalytic activity. These are transesterification reaction and most are concerned with metabolism. Ribosomal RNA hydrolyze amino acyl ester and play a role in peptide bond function. Example, peptidyl transferase activity. Important terms, cofactor. Molecule or ion that helps in catalytic reactions are called cofactors. Apoenzyme, the portion of the protein that needs cofactor for reactions are apoenzymes. Haloenzymes, enzyme as whole. Haloenzyme consists of apoenzyme and one cofactor. Enzymes, the protein that act as catalysts that speed up the reaction rate but do not alter the reaction nature. Coenzyme, the organic non-protein molecule that binds with apoenzyme to form active enzyme. Coenzymes are vitamins. Endoenzyme, a cell made enzyme and it is not secreted or exported out of the cell. Substrate, the specific molecule on which enzyme act. Metabolism, chemical reaction of the cell that are under control of enzyme. Catabolic reactions, the reaction in which chemical compounds are broken. Anabolic reactions, the reactions in which chemical compounds are synthesized. Enzyme immobilization, when an enzyme attached to the solid matrix and cannot move and act on substrate, it is said to be immobile and process is called immobilization. Enzyme inactivation, when an enzyme doesn't react or participate in activity due to inhibitor or inhibitory reactions, it is called enzyme inactivation. Example, in vivo and vitro reactions. Enzyme stabilization, the process of reducing the inhibitory conditions so that the enzyme can work properly. There are two types of enzyme, exoenzymes and endoenzymes. Endoenzymes are those enzymes that act inside the cell. Exoenzymes are those enzymes that act outside the cell. Enzymes are the protein may have non-protein component. It can be organic compound or metal. If it's an organic compound, it is called coenzyme, and if it is a metal, it is called cofactor. And the protein and non-protein part are called haloenzyme. When the non-protein part tightly bound to protein, it is called prosthetic group. When protein part dissociates from protein part, it is called apoenzyme. Nomenclature and classification of enzyme. Enzyme is named as ase by addition of ase at the end. For example, if maltose is getting converted to glucose, the enzyme name is maltase. International Unit of Biochemistry (IUB) has 6 major classes. There are code numbers assigned to the enzyme that were prefixed by EC. It contains 4 points separated by points. The first number shows the enzyme belong to which of the six main classes. The second figure shows the subclass. The third figure gives sub-subclass, and the fourth figure gives the serial number of enzyme. The fourth digit identifies the specific enzyme. Alcohol NAD oxidoreductase is given 1:1:1:1. Major classes of enzyme and types of reaction catalyzed. Oxidoreductase EC1. They help in oxidizing and reduction reactions. Example, dehydrogenase, removal of 2 hydrogen, or oxidase, reduction of molecular oxygen. The second class is transferase, also known as EC2. Transfer of group between two substrate. Reactions like aminotransferase that catalyze the exchange between amino acid and keto group between amino acid and keto acids. Hydrolase section EC3. They help in hydrolysis of substrate. Peptidase that catalyze hydrolysis of peptide bond. Lyase EC4. They help in the removal of the group from the substrate non-hydrolytically. Example, decarboxylase that catalyze the removal of carboxylic group from the substrate. Isomerase, it helps in the isomerization of substrate. Example, racemase that convert D isomers to L isomers and vice versa. Ligase is EC6. It belongs. Specificity of enzyme. Enzymes are very specific in their action. Enzymes have four types of specificity. One, reaction specificity. They catalyze only one type of reactions and act on specific substrate. Example, urea act on urease and catalyze on catalase on hydrogen peroxide. Bond specificity. These enzymes act on specific bonds, glycosidic or peptide bonds. Example, pepsin, trypsin act on peptide bonds only. Group specificity. These are group specific. They act on particular functional group. Example, alcohol dehydrogenase act on OH group. Optical specificity. They exhibit optical specificity for at least a portion of substrate molecule. Example, maltase catalyze the hydrolysis of alpha glycosidic bond between two glucose molecule but not beta glycosidic bond. Mechanism of enzyme action. Michaelis and Menten states that enzyme first bind to the substrate to form enzyme substrate complex, that is denoted by ES. Then it dissociate to form protein and unaltered enzyme. When the substrate binds on the active site of enzyme, the action of enzyme starts. The catalytic site of enzyme molecule possesses complex 3D form and provides a cleft that binds substrate. ES complex is formed by non-covalent bond between group of substrate and amino acid chain present at catalytic site. Models of ES complex formation. There are two models. One is lock and key method, or it is also called as Fisher model. It says the catalytic site has proper conformation compatible to substrate even if the substrate is absent. The catalytic site binds the substrate and catalyzes the reaction without any change in its own conformation. Failures. This model fails to explain the change of enzyme activity in presence of allosteric modulators. Allosteric modulators are those substances when they bind on the enzyme molecule other than the catalytic site, they either inhibit or enhance the activity. This model also fails to explain the action of non-competitive inhibitors. Koshland induced fit model. It says the catalytic site doesn't have a proper conformation as explained in lock and key method. When substrate approaches the enzyme, the conformation of catalytic site change according to substrate so that the enzyme holds its substrate properly. This model very well explains the non-competitive inhibition and allosteric modulation of enzyme. Cutter. It is the unit of enzyme activity. It is the amount of an enzyme that transforms 1 mole of substrate into product in 1 second. IU is the amount of enzyme that catalyzes the transformation of IU mole of substrate into product in 1 minute. Specific activity is expressed as KAT slash kg or IU slash mg. Molar activity is denoted as KAT slash mole. Turnover number of enzyme, number of molecule of substrate transformed per catalytic site of enzyme per minute. Thank you so much, and we will be soon coming up with the second part of enzyme and coenzyme. Stay safe, stay protected. Bye bye.